Molecular physiology is a branch of physiology that studies the function and interactions of biological molecules that underlie the activity of cells, tissues, organs, and body systems as a whole. This study focuses on molecules such as DNA, RNA, proteins, enzymes, lipids, and carbohydrates that play a role in the basic mechanisms of life. Here is a more detailed explanation of molecular physiology. 1. Definition and scope of molecular physiology. Molecular physiology explains how molecules interact and play a role in regulation of genetic activity, gene expression, energy metabolism in cells, the process of communication between cells through chemical signals, biological processes such as cell division, differentiation, and apoptosis. This study integrates molecular biology, biochemistry, and physiology to understand molecular mechanisms at the cellular and organismal levels. 2. Main components in molecular physiology. A. DNA, deoxyribonucleic acid, main function, store genetic information that regulates protein formation. Control the inheritance of traits. Important processes: DNA replication, DNA duplication for cell division, transcription, copying genetic information from DNA into RNA. B. RNA, ribonucleic acid, main function, directing protein synthesis. Being an intermediary molecule between DNA and protein. Types of RNA: mRNA, messenger RNA, carrying genetic templates to ribosomes. tRNA, transfer RNA, transporting amino acids for protein synthesis. rRNA, ribosomal RNA, forming part of ribosomes. C. Protein main function, being an enzyme that catalyzes chemical reactions, forming cell and tissue structures such as actin and collagen, being a signal molecule such as hormones and receptors. D. Enzymes main function, catalyzing biochemical reactions, such as the breakdown of glucose molecules in energy metabolism, regulating the speed of biochemical reactions without reacting. E. Lipids and carbohydrates. Lipids play a role in energy storage, cell membrane formation, and cellular signaling. Carbohydrates are the main source of energy and raw materials for the biosynthesis of other molecules. 3. Important processes in molecular physiology. A. Genetic expression. Transcription. DNA is transcribed into RNA. Translation. RNA is translated into protein in ribosomes. Genetic regulation. Transcription and epigenetic factors control when and how much protein is produced. B. Energy metabolism. The process of converting energy from organic molecules such as glucose into ATP. Adenosine triphosphate, the main energy source of cells. Main stages: glycolysis. Breakdown of glucose into pyruvic acid in the cytoplasm. Krebs cycle. Conversion of pyruvic acid into energy molecules in the mitochondria. Electron transport chain. ATP production in the mitochondria. C. Cellular signaling. Signal molecules such as hormones and neurotransmitters transmit information between cells. Signal transduction pathways. Signals are received by receptors on the cell membrane and translated into cellular responses. Example: insulin receptors regulate glucose uptake by cells. D. DNA repair mechanisms such as base excision repair and mismatch repair maintain genetic stability. E. Apoptosis, programmed cell death, a controlled molecular process to eliminate damaged or unnecessary cells. 4. Molecular mechanisms in body systems. A. Nervous system neurotransmission. Communication between neurons via neurotransmitters such as dopamine and serotonin. Ionic signaling. The flow of ions through ion channels generates nerve impulses. B. Endocrine system molecules such as steroid hormones, estrogen, cortisol, interact with nuclear receptors to regulate gene expression. Protein hormones, insulin, initiate signal transduction pathways to regulate glucose metabolism. C. Immune system immune receptors, such as toll-like receptors, TLRs. Recognize foreign molecules and activate immune responses. Antibodies, proteins that bind to antigens to fight pathogens. 5. Role in disease. Molecular physiology helps understand the mechanisms of disease at the molecular level. Cancer, genetic mutations that cause abnormal protein expression. Diabetes, impaired insulin signaling that inhibits glucose uptake by cells. Alzheimer's, accumulation of beta-amyloid protein that damages neurons. 6. Technologies in the study of molecular physiology. Genomic technologies, sequencing DNA and RNA to study genes and their expression. Proteomics, analysis of proteins in cells to understand their specific functions. Bioinformatics, using computers to analyze large molecular data sets. Conclusion: molecular physiology explains the molecular basis of all life processes. By understanding the molecular mechanisms, we can develop new therapies for various diseases, such as gene therapy, drug development, and molecule-based medicine.